Hello and welcome to a quick tutorial that's going to show you how to find unusual glycosylations in proteins using two tools. One is GLAM, and then the second one that we'll be using to do the actual searching of the mass spec data is called PGFinder. So, step one, make sure you've got a couple of things on hand already. So, most important things is that you have a FASTA file of the protein or proteins of interest. In our case, that's going to be the spike protein from SARS-CoV-2. So here you can see just a normal FASTA file with the sequence, nothing special about that. And the second thing you're going to need is a list of glycans that are then attached to the peptides from, or attached to that protein, I suppose. So this protein here was recombinantly expressed in Chlamydomonas, so it has an unusual set of glycans that we've described here in this CSV file. The important part is that you have a column called glycan, and then inside you're giving a composition of each of the glycans. Or you can give the Oxford notation, or even the full IUPAC notation for glycans. There's a couple of different things that are accepted by GlycoWork, which is the backing library for this. Additionally, if you want to give things arbitrary names and you already have masses calculated, you can add another column over here that says monoisotopic mass with a space like that and capitals. And then here you can go ahead and give it a list of masses, and then it won't try and calculate them automatically from this anymore. It'll just use the number that you give it. But for our case, we're going to keep it with the automatically calculated masses, and here's just, listing the composition of those, the number of each sugars, 4 hexes, 2 hexnacs, that kind of thing. Cool, so now that we have worked that out, there's one last thing you'll need to make sure that you have eventually. We won't need it for this first step, which is building the database, but we will need it when we're actually trying to search our mass spec data, and that is some sort of deconvoluted mass spec data. This is a file that's been generated by BIOS. You can also generate it with open source software like MaxQuant. What's important here is just that you've pulled out from your mass spec data some list of ions and charges that can be used by PGFinder. So PGFinder's website has more detail on how to generate these sorts of files. Right, so let's get started with actually generating this database. First, we'll jump back over to GLAM, and you can see that this is split into 3 different steps. You have a step where we're generating different peptides from our protein, a second step where we're going to attach glycans to those peptides, and then a final step where we're going to be building a database that can be used in PGFinder. So, first of all, let's upload our algal spike protein. Here we are, you can see down here the protein's been uploaded, and we can move on to our next step, which is specifying the digests. So this will depend on how you've prepared your data for mass spec. I know that this data set was generated via a triptych digest, so I'll scroll down and select trypsin. If you want to select several treatments, so say you've done a co-digestion, like with trypsin and elastase, for example, you can hold Control and click it, and now elastase and trypsin will be selected. You can also do things where you shift-click to select a range of things. Just be aware you can pick multiple digestions. In our case, though, we're only interested in a single digestion with trypsin, so we can select that. Note that if you don't have a digest that's on this list, you can also put in a custom regular expression rule that describes the cut. That's a more advanced feature that we won't dive too deeply into today. But just know if something's missing from this list, you can still simulate a digest with it if you know its cutting rule, I suppose. Next, we'll open up advanced options here, and you can see a couple of things. One is a helpful one called missed cleavages. So currently, if you select a digestion like trypsin, it will only generate peptides that have been cut at every single trypsin site, I guess, if that makes sense. So you'll never have a potential trypsin site in the middle of your peptide, because if it were there, it would have been cut, and so that peptide wouldn't exist, kind of thing. If you increase the number of missed cleavages, you can get a larger number of peptides that get longer. So we'll do that so we can see a couple more things when we do our search later. And you can set a minimum and maximum peptide length. Here, I don't know, I'll put it down to 5 amino acids and then make it up to 40 amino acids. You can configure these obviously however you want. Now, one particularly weird option here is the semi-specific cleavage. That's not too important for now, but just know that this option essentially will take your peptides generated by the triptych digest, and then it will go ahead and remove all of the prefixes and suffixes from that peptide. So by turning this on, you're essentially taking your peptide, which has been cut once already out of the protein, and you're cutting it again at every individual cut site. But we'll keep that off for now, keep our database small, and move on to the next step, which is specifying modifications. So over here, you can see we have a number of different modifications. It's the same as before, where you can either control click or you can shift click to select multiple of them. And you can also go over here to this custom tab to see a bit more information about them and to add new ones of your own. Important thing to know is just, this is the abbreviation, important that it's in lowercase, that describes in the final peptide sequence which residue is modified. So you see ox and then a capital M for an oxidized methionine, for example. And then the target residues, you can have several of these, you just have to comma separate them. So you can do something like that. And now this modification would target 2 different residues instead of just 1. Now, for now, I'll go ahead and just go back to the built-in ones, check that that's reset our custom things, and open the advanced options. Here there's another tool for limiting the size of your search space. So obviously you can imagine as peptides get long, there's lots of different combinations which you can put different modifications on them, and you end up with this kind of combinatorial explosion of complexity. So if we want to keep things simpler, we can limit the number of modifications that can be put on a single peptide. So here I'll put a rather small limit of just 3 modifications on a single peptide. So if a peptide has like 4 different modifications, we might miss it, but it's keeping our search space small to start. You can expand these things as you do more targeted searches later, I suppose. Cool. Moving on to step 2 then, attaching the glycans. So this one's a bit simpler than step 1. First thing we want to do is upload our glycan file. So there's our CSV file from earlier. And then we can select the glycosylation motif. So this is where the glycan is attached. Currently built in is just in glycosylation, but we can add other types of glycosylation if that's something you are interested in. But do know as well that there's this escape hatch. You can put in a custom regular expression in the meantime. Okay, advanced options for glycans. You've got 2, both of them kind of restricting the search space or expanding it in different ways. So by default, if a peptide has, say, 3 different glycosylation sites, then it will generate peptides with every combination of glycosylations for that, if that makes sense. So if all 3 sites are glycosylated, then it will be every possible combination of all 3 of the glycans in your database. It will also generate ones for if only 2 of the 3 are, or if 1 of the 3 are, things like that. This adds quite a lot of complexity to your database very quickly. So we'll put a smaller limit, say just 2 glycans can be attached per peptide, and we'll see what that looks like in our result file later, but keeping that small prevents this from taking too long to generate. Finally, this setting here lets you decide whether or not your database file should include the peptides from step 1, or if they should all be filtered so it only includes the peptides that have glycans attached to them, or rather, I guess, have this glycosylation site within them. Sometimes helpful to flip this on if you want to see in general, has your mass spec data collection managed to catch lots of these peptides, even if it's not caught any glycans. It's a good quality control check, but for our case, to keep our PGFinder search later simpler, we'll keep this setting off. Finally then, we get to our build database step. That one's pretty easy. You press the big build database button, takes a moment to generate, and then you can see something's been downloaded onto our computer. From here, we can jump over to the mass spec tool, which is actually a separate website, PGFinder, but it's helpfully linked. And then here, we're only concerned with this MS analysis module right in the middle. So first step we want to do is upload our mass spec file that's been deconvoluted. So here's that features file from earlier that I mentioned. You want to ignore the built-in databases because this is for peptidoglycan, but we're interested in glycopeptides here. Go over to this custom mass database, and then upload that one that we just generated. So I'll go over here, take that file we just generated, and then I'll open advanced options to show you what's here. Most of this stuff is relevant only for peptidoglycan, so we can ignore quite a lot of it. But this tolerance setting is generally helpful, so you can widen or tighten your tolerance depending on the accuracy of your mass spec instrument, and things like that. By default, it's set to 10 ppm, which is a pretty tight tolerance, but that should work fine for Orbitrap instruments. So now I'll go ahead and press Run Analysis, and we can see PGFinder starts thinking, analyzing the data, and I'll go ahead and pause things here until the data analysis finishes, and then I'll show you what the final output file should look like. Alright, welcome back. You can see it's finished processing down here, and it's downloaded onto our computer a results file. So we'll go ahead and take a look at that. We'll pop over to our downloads, and we will see that there's this new file here. if we go ahead and open our output file, it should be named, I think, the same thing by default as your dataset will be. Going in, cool, and we can zoom this out just a little bit. This is a normal PGFinder output, so it's got some basic information like the retention time of the ion, its charge, and its observed mass. And then next to it we have the theoretical mass, so this is the thing that was calculated by our GLAM tool. This is the, predicted monoisotopic mass. And then here's the delta ppm, so that shows us how much error there is, essentially, in this, between the theoretical and actual observed masses. And then finally, of course, this is the most interesting column. It has our big list of inferred structures. So here you can see, for example, up here, a decently large peptide that has one type of glycan on it. This one's quite abundant. You can see this intensity column telling you it's done an integration of the extracted ion chromatogram, the XIC. So higher intensity means a more abundant structure, typically. This then is our most abundant glycopeptide, but there's lots of others around. This one, for example, is an interesting one because there's this little plus symbol in the middle. So that means that there are 2 glycans attached to this particular peptide, because this peptide has 2 separate glycosylation sites. So if we wanted to check out in more detail, for example, where those glycosylation sites are, and where this peptide comes from in the overall protein, we can do something like take this structure, copy it, and then we can look back at our original database, which will have a bit more of that supplementary information. So I'll go ahead and open up this one here, and then do a quick search of this database to find that structure. And ta-da, here we are, we can see it. Down here then, you can see that this peptide begins at position 336 in the protein and runs out to 353, and it has 2 different glycosylation sites, one at position 338 and one at position 350. So that's a way you can get a little bit more information. And then this mass is the same one as that theoretical mass in PGFinder. And you can also see there's quite a lot of things in this particular database. That database that we just searched was a massive one. It had 148,000 different structures that we just looked for. So that's a nice quick demo of PGFinder and the kind of things you can get from it. You can see there's quite a number of predicted glycopeptides here. Now, of course, it's worth confirming these things with, MS2 and looking at the fragmentation, other things like that, taking a look at the error. Some of these are obviously a lot closer than other ones might be in terms of the delta ppm, so that's worth keeping in mind. But this is a great first place to start, and it kind of gives you an idea of what might be in the data set that you can then dig deeper to confirm. Hopefully that's been a helpful demonstration of both GLAM and PGFinder, 2 tools that are freely available online. If you have any issues with either of them, please feel free to reach out to the contact email at the bottom of both pages, and we will try and get back to you and help you out with whatever issues you might be facing. Awesome. And of course, if you find this tool useful, feel free to cite us. Cool. See you. Good luck. Enjoy finding some glycans.